Hello friends, I am Ayantika and in this video I will be discussing about Chlamydomonas as a model organism in research. So in this video I will be discussing how Chlamydomonas is used as a research tool in molecular biology, in the ciliary study, the chloroplast dynamics, in forward and reverse genetics, in optogenetics, in understanding the evolutionary process in algae, in biological hydrogen production, in applied biology, understanding the morphology and function of organelles, the intracellular organelles, and the various biosynthetic pathway that happens. Now, this model organism, what is a model organism? A model organism will be an organism such that it has a small size, a very due to the small size, it will have a simple structure. It will be haploid. So it can also have a very high generation speed, that is its life cycle is completed in a very short period of time. It has a very fast growth rate actually. So, and also the model organism should be very easy to culture and store. That is, it will be requiring minimum amount of resource to culture and store this organism, and also it can be stored easily. Stock can be maintained easily, and main quality, one of the main quality required for a model organism is that it should be easily genetically manipulated. It can be easily genetically as because it can be genetically manipulated, it can be used in reverse genetics and applied research. So all these are the characteristics that will be required for any organism to be called as a model organism. Now, this all these characteristics are shared by many organisms, and we have different model organisms in our research. Say for example, Escherichia coli, the bacteria that is present in our intestine, that is also used as a model organism. And bacteriophage, that is the virus that infects the bacteria. So this bacteriophage, and again the worm Caenorhabditis elegans, and the mold yeast, that is a Saccharomyces cerevisiae, our garden pea Pisum sativum, the corn, that is a Zea mays, and another plant called Arabidopsis thaliana. These are all model organism for study and research. Also, if we consider the animals, then Drosophila melanogaster, that is a fruit fly, is a very common model organism, along with Mus musculus, that is a mouse, and another is the zebrafish, that is the Danio rareo. So we see that for the research work, there are many model organisms, and all these organisms, they share the common characteristics due to which they are all called model organisms. So here we will be discussing what are the characteristics that make Chlamydomonas the green algae that was discovered by Christian Ehrenberg, and what are the characteristics that make it a good model organism, and how these characteristics are helpful in various research work. So Chlamydomonas, it is because it is a haploid organism, then any mutation will be rapidly seen, because because it is a haploid, and its life cycle is very short. So if there is any mutation, it will be easily observed in the very next generation. So, being a haploid organism, it is very good in the study of either genetics or in studying the biosynthetic pathway. So, if there is any mutation, there can be loss of function of certain genes, which can in turn be causing mutation, loss of certain proteins. So, this being a haploid organism, it can be the phenotypes, as they are easily expressed, it can be used in the study of both genetics as well as for biosynthetic pathway. Now, Chlamydomonas, it grows very rapidly. That is, it has, and also the generation time is just 8 hours. So we see that it has a very short life cycle of 8 hours, and among all of the Chlamydomonas species, this Chlamydomonas reinhardtii is the most commonly used model organism. Also, some species of Chlamydomonas, they are thermophilic, which means that they can grow in high temperature conditions also. And we know that for surviving in the high temperature, any organism surviving in high temperature, they require thermostable enzymes. This thermostable enzymes. So this property helps the scientist to commercially use it, because the thermos, there are certain so many various processes, manufacturing processes, where high temperature is very difficult, low temperature is very difficult to maintain. So temperature rises due to certain reactions, but this thermostable enzymes will thus help in those reactions where it is very difficult to maintain a low temperature. So in high temperature also the reactions will happen. So this property, or the thermophilic property of this algae Chlamydomonas, is widely used in this type of commercial processes. So Chlamydomonas and can also survive in the hypoxic or oxygen containing environment. So that is, it means that it can grow in both oxygen as well as non-oxygen containing. In oxygenic condition, it have glycolysis, and in non-oxygen containing, that is hypoxic condition, it does survive by fermentation. So under both oxic as well as hypoxic condition, Chlamydomonas can survive, and this property can be used to study the various biosynthetic pathways in under hypoxic as well as oxic condition. So Chlamydomonas can be induced to reproduce sexually by creating an artificial shortage of nutrients, especially the nitrogen containing nutrients. In absence of this nitrogen containing, in shortage of this nitrogen containing nutrient, Chlamydomonas shifts from the asexual mode to the sexual mode of reproduction, and if there is any mutation, and how this mutation is carried on further, and how this type of mutation will affect the organism, or if the mutation is of any commercial use to us or not, that can be easily understood by introduction of mutation and induction of the Chlamydomonas for sexual reproduction. And by this process we can see that not only a single gene, but multiple traits can be mutated, and we can easily obtain the result of the mutation in the very next generation. So this is the process of reverse genetics, and mutation can be done both by chemical mutagens as well as physical mutagens, I mean the gamma rays, and as well as there can be gene disruption by using this CRISPR-Cas method. And by this way, not only the effect of mutation is studied, but also in forward genetics also, for the construction of genomic library, it is very useful. In optogenetics is basically the field of genetics that deals with light activated proteins. For example, in Chlamydomonas reinhardtii, which is the model organism for optogenetics, uh, uh, the 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 eye spot, it contains channel rhodopsin. Eye spot, that is a orange colored or brown colored body that is present in the anterior region of the cup shaped chloroplast. It contains channel rhodopsins, which are the ionic channels that are activated by light. Now this channel rhodopsin 1 and channel rhodopsin 2, that is isolated from Chlamydomonas reinhardtii, these are seen, this type of when they are isolated and the gene isolated, it was seen that if this gene is introduced in other organism also, they can produce the same type of light gated ion channel response. This channel rhodopsin 1 and 2 are basically channel rhodopsin 1 and 2. These are basically light gated ion channel. These light gated ion channel can be used in the study of similar processes that happen in other organism also, that is especially in our human eye. See, it is very easy to study this type of any research work on a single celled organism than a complex organ containing organisms like us. We have many organs, and it is very difficult to study in a multicellular organ or organism. So this study of light gated ion channels in a single-celled organism is much more easy to research and study. Chlamydomonas reinhardtii also used in the production of biological hydrogen. Uh, Due to under certain circumstances, it stops producing oxygen. It turns to a different mode of photosynthesis using not water but H2S, where by use of this hydrogenase, hydrogen is produced. So this chlamydomonas can be used in the production of biological hydrogen. I have made a video regarding the biological hydrogen production, how biological hydrogen production is done using various algae. So if you want to know about the detail of how this entire process is done, you can easily go to that video, biological hydrogen production. I have put the link in the description box. So I will not be discussing it in detail how the entire process is done. Now the applied biology. Applied biology is the use of organisms for our benefit, for industrial production of certain things that are beneficial for us, means that will be needed by us. So this chlamydomonas, as it has got we know all the genomic sequences of the chlamydomonas there from the gene library. It has got an elaborate genomic structure, and we know the biosynthetic processes that are happening here, so we can easily manipulate this chlamydomonas for the production of human proteins, especially for example this human antibody protein, the growth factors, that is the vascular endothelial growth factors. So all these various vaccines, especially the edible vaccines again different type of viruses, and many type of edible drugs that are used to treat cancer. So all this manipulation can be done, and chlamydomonas can be thus used in applied biology also. Chlamydomonas can also be used, as it is a very primitive organism. It can be used as a model for studying the evolutionary trend in algae. The flagellar microtubules of Chlamydomonas, they have a similar protein composition to that of the eukaryotic cilia or eukaryotic microtubules. So, by the study of Chlamydomonas, how the organization of the Chlamydomonas cilia, their the Chlamydomonas flagella, their assembly, their disassembly, we can easily understand diseases, human diseases, especially human body. It has got cilia in the ventricles of the brain, and in the inner respiratory tract, in the kidney also. But and also this entire study can be also used in understanding the male infertility related problems. So as the protein composition is similar, so by just the study of uh, the assembly, the disassembly, and various other processes related with the microtubules of the flagella, we can get to know about the diseases like polycystic kidney disease, any brain related disorder, any respiratory disorder, or disorder related to male fertility. All this can be used, and especially any mutative study in the Chlamydomonas reinhardtii can be done to understand the effects of various genes that can the effect of mutation of various genes that relates to various defective ciliary problems. And this process can help in understanding what mutations are responsible for the dysfunction ciliary functions in human being. See, the Chlamydomonas, it has got it is a prokaryotic cell. So every structure, the cellular structure, the biosynthetic processes that happen in the cell can be easily used to study the biosynthetic processes that are happening in us and us human, and also to understand the ultrastructure of various organelles um, in human being as well as other animals also. So this intracellular organelle study and the study of the intracellular functioning of various organelles are also of use in lab. The electron transport, the photosynthesis and electron transport in Chlamydomonas can also be studied to understand photosynthesis and electron transport that happens in plants. As because it is a single celled organism, it is very much easier to study the various photosystem related processes, various proteins and various photosystem related and the electron transport system easily than in a multicellular plant body, because this is a single celled, and also algae being a thalloid organism, it is much more easier to study photosynthesis related processes in this single celled organism rather than in many celled plants. So with this I come to the end of the video. This was all about the Chlamydomonas, how it is used as a model organism. Apart from Chlamydomonas, Volvox, which is also a blue-green algae, is also used as a model organism for study. There are also many other type of microbes, especially E. coli, bacteriophage, and Saccharomyces cerevisiae. These are also used as a model organism for various research purposes. If you want me to make a video on this other organisms as how they are used as a model and why they are used as a model organism, please write it in the comment section. Thanks for watching this video, and if you like it, share it with your friends, and do subscribe to get the latest videos that I made. Thank you for watching.